Part 3 is going to focus on the physiology of the digestive tract. I'm going to do this lecture by taking you through the digestive tract and just hitting on any physiology that was not mentioned in the anatomy lectures. So the control of digestion is mainly by the autonomic nervous system, which is the automatic nervous system, right? So you don't control the movements by that. The sympathetic nervous system inhibits digestive actions, while the parasympathetic nervous system promotes digestive actions. And this has been different up until this point, because when we have talked about like the cardiovascular system and the respiratory system and the blood vessel vasoconstriction and dilation, all of that was stimulated by the sympathetic nervous system, which is fight or flight. Well, now the parasympathetic system, which is the rest and digest system, is going to be in effect. The only thing that the sympathetic nervous system really does for digestive is to contract sphincters until it's time to allow contents to move through them. But basically, this is going to focus a lot around the parasympathetic nervous system. Hormone secretion is very important in digestion and gastric motility, actually digestive motility in general. Different enzymes are going to be released along the way. Different pHs are going to occur along the way. So the pH of like your mouth and your esophagus is more of a neutral pH. Your stomach drops down to a pH of about 1 to 2. And then the neutral pH resumes in the small intestine. So it's really important that we have that pH control on either side of the stomach through the alimentary canal, because otherwise different enzymes and things would not be able to work. And basically the presence of peristaltic movements is going to drive the next step to happen. So if that wave is detected in the next step, then that next step will begin to do what it needs to do. So beginning in the mouth, salivation is controlled by both sympathetic and parasympathetic, mostly parasympathetic. But if we are talking about the sympathetic nervous system, you might start to salivate if you are stressed out, scared, or have increased your physical activity. That's your fight or flight, right? Your rest and digest innervation is going to be from seeing food, smelling food, tasting food, even thinking about food. You might just start to salivate. And remember that a major enzyme found in the saliva secretions is salivary amylase, which begins carbohydrate breakdown. Just to kind of review that swallowing mechanism, this is from lecture 1. Recall that when the food is in the mouth, the tongue and palate kind of force the food bolus back to the pharynx, and then we can initiate those constrictor muscles to contract back there and initiate that swallowing down the esophagus. And remember that this esophagus, the first part of the esophagus, the superior portion, is going to be voluntary. And then as the food moves down the esophagus, that action will become involuntary, and peristalsis will take over and push that food content to the stomach. In the stomach, the major enzyme here is going to be pepsin. And when you see pep, you think protein. Interestingly, at this point with the stomach and then some pancreatic enzymes, they're actually inactive until they're activated. So for example, the stomach has something called pepsinogen, which is the inactive form of pepsin. When that mixes with the hydrochloric acid, which is in the stomach, then that activates it into pepsin, which then can break down that protein. So it's important that the acidic environment from the HCl is present, because if not, then this enzyme would not be able to work properly. So there's secretory cells within the wall of the stomach that secrete these enzymes and the hydrochloric acid. All of that combined, we call it gastric juice. Other things that are secreted by the stomach: gastric lipase. Lip or lip means fats. And in the previous lecture, I did mention that fat breakdown occurs and finishes in the small intestine. Only about 10% of fat is broken down in the stomach. So it's quite insignificant, but there is gastric lipase that's available. Intrinsic factors, very important for B12 absorption. B12 is necessary for red blood cell function. And then also there are some appetite regulating hormones, ghrelin, leptin, things like that. That I believe is all in the nutrition and metabolism chapter that we will not be touching on in this class, but that chapter is available for you if you're interested in those types of topics. So gastric secretion regulation. Okay, so there's a negative feedback mechanism happening here with the parasympathetic nervous system. Essentially, if we need to inhibit acid secretion, so let's just say that our stomach is empty, right? We don't want to secrete acid if there's nothing in our stomach to break down. So somatostatin is going to inhibit the acid secretion when our stomach's empty. Now, when food contents enter the stomach, then acetylcholine is released. What that does is turns off the somatostatin, therefore activating the secretion. Okay, and I threw this in here, do you remember how this had an effect on the heart, acetylcholine? Recall that triggered the heart to slow down, right? And that basically was a function of the parasympathetic nervous system. And so now we're making that connection of acetylcholine being very active in parasympathetic responses in terms of cardiovascular, which would have been the slowing down of the SA node firing, and stimulating digestion to happen by increasing gastric secretions. And, you can't have a fast heart rate and digest at the same time. I mean, you could, but that's not really how the nervous system works. Gastrin is another hormone, and aside from appetite, which is in that nutrition and metabolism chapter, this is also going to stimulate the cell regeneration in the gastric wall, in the stomach wall. Also, just basically increase gastric secretions. When it releases histamine, that increases it. So this is just an image from your book. Basically, a parasympathetic nerve impulse comes down to the stomach, targets that. That is going to stimulate the release of juice in the stomach and gastrin into the bloodstream, which is a hormone. And then gastrin is going to further tell that stomach to keep producing gastric juice. So now you have kind of a positive feedback type mechanism in which we are continuing a trend, right? We're releasing even more juice in response to the juice that's already been released. Gastric secretion can be broken up into 3 phases. So we have the cephalic phase, the gastric phase, and the intestinal phase. The cephalic phase is before your food even reaches the stomach. It might even be before the food enters your mouth. This is when you see the food, you think about the food, your stomach is preparing to eat. Maybe your stomach is growling, something like that. The gastric phase happens when that food actually enters the stomach. So you've eaten the food, swallowed the food, and now that food is entering the stomach. This is when all of those secretions are going to happen during that gastric phase. Once those food contents exit the stomach and enter the duodenum, now we're in the intestinal phase. And once this happens, once the stomach empties, now we need to turn off those gastric secretions. Otherwise, our stomach lining could be damaged by the excess enzyme and hydrochloric acid secretion. And that is going to be by something called cholecystokinin or CCK, and then also somatostatin is going to inhibit that as well. So what does the stomach do other than churn, right? It's going to absorb very small volumes of water, salts, lipid soluble drugs as well, and alcohol. Nutrients are not absorbed in the stomach, if at all. As the stomach expands, stretch receptors are going to be stimulated, and that is going to tell the peristaltic waves to push that chyme towards the pyloric sphincter, as well as contract that stomach, mix the contents, things like that, prior to pushing it towards the sphincter. But as that stomach expands, it distends. That's going to stimulate stretch receptors. That is also going to trigger a cascade in the small intestine after that as well. And because remember, we're all about preparing for the next step. And so as that stomach distends, the stretch receptors tell the next step to do something in response. Peristaltic waves are going to push that towards the pyloric sphincter, and that will eventually relax and open, and the stomach will empty. During all that, remember that the lower esophageal sphincter needs to close right after those contents enter the stomach. If it doesn't, the stomach starts churning, those contents can creep back up into the esophagus. That's where you get your heartburn and indigestion feelings from. Think about pH when you think about that. So the pH of the stomach is about 1 to 2. The esophagus was pretty neutral, around a 7. So if you have acidic contents going into your esophagus, that esophagus lining is not equipped to handle those acidic conditions. And so you can really do some damage to that tissue. How fast the stomach empties depends upon what you've eaten, essentially. And basically what happens here is, let's see. Okay. So the stomach empties, and that is the introduction of chyme into the duodenum or the small intestine. That's going to stretch that small intestine. That will tell the nervous system to basically inhibit these secretions and inhibit this peristalsis. If the stomach emptied too fast, peristalsis would have to increase here. And remember that the small intestine is necessary for absorption, and we need time to absorb those nutrients. So chyme has to enter the small intestine very slowly. If it goes too fast, then all of these contents are going to pass through the small intestine, and a lot of it's not going to be absorbed. So peristalsis is slowed, especially for fat digestion, because remember that hardly any of it has been digested up until that point. So less parasympathetic impulses, so less peristaltic waves, and an inhibitory hormone that helps with that is that CCK. The small intestine releases enzymes and mucus mostly, peptidases, which are going to be protein, intestinal lipase for fats, and then all of these are going to be carbohydrate enzymes to finish up that digestion and absorption mechanism in that small intestine. And basically these things are released once things enter the small intestine. Basically, it is based upon stretch receptors, but it's also based upon the pH change that it senses. And so our parasympathetic impulses are going to increase at that point, and we're going to have these things all released at that moment. Then remember that your parasympathetic impulses need to kind of slow down a little bit to allow that stomach to empty at a slower pace. So just to kind of recap, carbohydrate metabolism begins in the mouth and then completes in the small intestine. Protein began in the stomach, completes in the small intestine. Interestingly, amylase, which digests carbohydrates, is inactive at low pH. And so this begins in the mouth. It does not occur in the stomach. It resumes in the small intestine, which is kind of interesting. Protein will begin in the stomach, complete in the small intestine. Lipid nearly entirely in the small intestine, a little bit in the stomach, not much. Also, the small intestinal wall will absorb electrolytes by facilitated diffusion, and then water by passive osmosis. Oh, this picture was, just showing that lacteal here and those fatty acids being absorbed, those dietary lipids being absorbed into the lymphatic system. Lymphatic system will meet up with the bloodstream, remember, at the subclavians. Okay, so how fast does this go? Well, the small intestine contracts by segmentation as well as peristalsis. So it kind of has some mixing movements because that digestion has to complete first before things can be absorbed. Then it will move things through peristalsis, all driven by parasympathetic impulses. There is a stomach distension reflex, which triggers the small intestine to be receiving those contents. And then also peristalsis triggers sphincters to relax. So sphincters are usually constantly contracted and closed due to the sympathetic nervous system. But Peristalsis, which is triggered by the parasympathetic nervous system, relaxes those sphincters so things can keep moving forward. Remember that into the small intestine, we have pancreatic secretions and bile secretion. Pancreatic secretions, enzymes mostly, some bicarbonate, some nucleases, amylase will be protein, carbohydrate digestion, lipase will be fats, and then the last three are going to be protein. Amino acids need a lot of different types of enzymes to fully break them down. Nucleases are going to break down nucleic acids. And so basically now we're covered. So all of that stuff is going to be released into the small intestine, and all of the macromolecules should be able to be digested completely and then absorbed. Also, we have bicarbonate ions. Bicarbonate is a buffer. So remember that the contents from the stomach is very acidic. The small intestine pH is about neutral. So we need something to buffer that pH, bind up those hydrogen ions, to neutralize that so that we're not only damaging the small intestine wall because it's not equipped to handle those pHs, but also so it doesn't denature our enzymes that are there. Those enzymes don't necessarily work at low pHs. And so we don't want to denature those enzymes so that we can't digest anything. The parasympathetic nervous system again stimulates the pancreas to release those enzymes. Two different things released here, secretin and cholecystokinin, or CCK. Secretin responds to the acidity of the contents of chyme. And so secretin is going to release that bicarb from that pancreas. CCK is going to trigger those pancreatic enzymes. And basically in response to the detection of protein and fat in the small intestine. It says, hey, there's protein and fat in here. We need these enzymes to be able to break those things down. And this is kind of what this looks like from your book here. So we have acidic chyme entering the duodenum. That is going to stimulate secretin into the bloodstream because that detects the pH. This pH might be 1 or 2. The pH of the small intestine is about 7 to 8. So that doesn't match up. Secretin is released into the bloodstream, which is a hormone that stimulates the pancreas, targets the pancreas to secrete bicarb. And then that will come out through the pancreatic duct through the hepatopancreatic sphincter. So that's the secretin phase. Remember that not only are pancreatic enzymes released through here and bicarb, but also we have that bile that's released as well. So we have bile, which consists of bile salts, pigments, cholesterol, and electrolytes. Those are released also in response to CCK. So secretin, pH, right? CCK is going to be all the enzymes in the bile. That is going to tell the gallbladder to contract. So this gallbladder is ideally full of bile. When it contracts, it's going to squirt that bile through the common bile duct and then out through the hepatopancreatic sphincter. CCK is released in response to fat and protein in the duodenum. Remember that CCK also triggered pancreatic enzymes to be released. So now we have our enzymes, CCK is being released. Now we have our enzymes coming this way. We have our bile coming this way, and the sphincter is going to open due to peristalsis contractions that are happening here. So that's going to relax the sphincter, and those things are going to enter. Remember that fats need to be emulsified or broken down into smaller pieces prior to lipase enzymes breaking down the fat into fatty acids. Bile is also reabsorbed in the small intestine after this is all complete. Here's what this looks like. So here's your chyme, and this is the CCK mechanism, whereas this one was the secretin enzyme system. So here's CCK. So again, simultaneously as secretin's being released, we have that chyme entering the duodenum. It has fat in it, it has protein in it. Our cells detect that, the presence of that, and that's going to release CCK into the bloodstream, which is going to target the gallbladder to contract and squeeze the bile out. And then also the enzymes from the pancreas will also be excreted here as well. The large intestine doesn't do a whole lot in terms of function, and it's also very slow compared to the rest of the path up until this point. its main secretion is going to be mucus, and the parasympathetic impulses increase that mucus secretion. That mucus is important for pH control, but also kind of the binding of the fecal matter into a mass. It's going to absorb water by osmosis, electrolytes by active transport. Peristalsis occurs a lot slower than it does in the small intestine or stomach, only about 2 to 3 times daily. And then bowel movements are going to occur. So the defecation reflex basically occurs as those feces travel through the colon and then end up at the rectum. That's going to put pressure on the rectum, and that's going to increase peristalsis in the descending colon, which is right before the rectum. That's going to further push the contents this direction towards the exterior. That force is going to relax the internal anal sphincter, which is here. That is an involuntary sphincter. So that one is going to relax. That's going to be your urge or your pressure. And then the sphincter that you have control over, which is a larger sphincter, the external anal sphincter, that one's voluntary. And that's when you are ready to have a bowel movement. That one will relax and then you can defecate. All right, so that is all the physiology that I need you to know for the digestive system. Your book does go into a lot more detail. I would expect, as always, for you to read those chapters. And if you have any questions, just let me know.